Today we'll have a presentation about joint work that focused on quantum-aided drug discovery. I'm Hrant Garibian. I'm the co-founder and CEO of BlueQubit. BlueQubit is a quantum software company specializing on quantum simulators, quantum algorithms. And then today's presentation will be done by Haik Tepanian, CTO and co-founder of BlueQubit, and Bill Shipman, CTO and co-founder of PolarisQB. PolarisQB is a company focused on quantum-aided drug design and discovery. And this is a work supported by DARPA. And then Haik and Bill will share some of the exciting results. So I wanted to also open up, Haik, thanks for sharing the slides. And we also have a Q&A button, so feel free to add your questions in the Q&A section, and then in the end, we'll leave 5 to 10 minutes to go over the questions. So welcome again to this webinar, and looking forward to the discussion at the end. With that, I'll pass the torch to Haik to start out the presentation.
Thanks, Hrant. Hey everyone, I'm Haik, co-founder and CTO at BlueQubit. Today, me and Bill will be covering these areas. Basically, I'll start with a few words about where quantum computing is now in the world. I'll say a few words about what BlueQubit does and how we fit in that big picture. And then I'll pass the torch to Bill to talk about their use case and what Polaris did in partnership with BlueQubit using our platform and what use case they have worked on. So we are in the quantum computing world, and we are seeing that we've been monitoring the trends for a long time, and we are seeing that quantum adoption is growing pretty fast. And it all kind of kickstarted since 2019 from Google's quantum supremacy paper. More and more Fortune 500 companies started creating quantum teams and looking into different use cases of how quantum computing can solve their hardest and most challenging problems. And we are seeing that as quantum computers are getting bigger and more stable and more production ready, more and more enterprises are thinking about different ways to use these computers to approach their hardest problems. We ourselves are a quantum software company, BlueQubit, and we have been using quantum computers a lot to approach different problems and devise different ways to solve classical problems with quantum computers. And we ourselves seen this evolution when some 4 or 5 years ago, there were only 3 to 5 qubits available for people, and even those were very hard to get access to. But then later, we started getting to these computers that you could actually run very small toy examples. We ourselves built this algorithm called data loader that allows to prepare classical states in quantum computers, which is basically the first stage in any quantum algorithm. First, you need to input your data or your problem somehow into a quantum computer. So in 2023, we were able to prototype these first algorithms on 16 to 27 qubits, still very small. This quantum computer still could be simulated perfectly inside a regular laptop, so nothing major. But last year, basically 2024, we started getting to this regime where now we are dealing with hundreds of qubits. We can run thousands of 2-qubit gates, which are the equivalent of operations on classical computers, with the difference that quantum operations are much, much more versatile, and thousands of CNOT gates basically mean that we can run things on quantum computers that are on par with classical computers on specific use cases. And moving forward, 2025, this year and next year, we're expecting to see a lot of what we call scientific quantum advantage, meaning use cases that might not be commercially valuable yet, but are still examples of problems where quantum computers can perform a computation or solve a specific problem in a way that cannot be done on regular computers. Based on that, we've seen that large enterprises and Fortune 1000 quantum companies have a strategy where they basically start a team or have few people dedicated to quantum who are kind of looking into application discovery, right? The first stage is to understand what problems they can even approach with a quantum computer. And because quantum computing is a totally different paradigm, it's not like going from one uh, language like C++ to Python or something. It's a totally different paradigm. You need a totally different thinking for this. That's why it's really non-trivial to do market research or even just internally understand where quantum computers could be useful inside your organization. And when doing this application discovery, the results of how good your discovery is basically depend on the quantum hardware that you have access to and the quantum software and algorithms that you have access to. And that's what we do at BlueQubit. Basically, this is how we fit in the big picture. We basically have a platform that provides access to quantum computers, to simulators, NVIDIA powered GPU accelerators, and it's the easiest way to run any quantum program and prototype any quantum algorithm. And we have all kinds of simulators and all kinds of tools for cutting edge quantum research. Um, PolarisQB is one company that's been using our platform for chemistry use cases. So I'll pass the torch to Bill now, who will be basically presenting what they did with our platform and what use cases they are looking into.
Awesome. Very good. Thank you very much. So, Bill Shipman, CTO and co-founder of Polaris Quantum Biotech, want to really thank BlueQubit for sharing the platform and webinar with us today. It's a great opportunity to talk about the work we've done together. So for a quick soundbite on who Polaris is, we were founded in Durham, North Carolina in 2020. Half our team are computational chemists and half of them are software engineers. We like to describe what we do as translating chemistry into the language of quantum computers, because as Hayek said, it's a radically different paradigm, which means you really need to reconceptualize the problem that you want to solve. And perhaps there's a different problem that's equally valuable that can get you to functionally equivalent results that isn't the problem you would necessarily think of a priori. And I'll get into that a little bit more. Let's see, are the controls working? Yay, they are. All right. So sorry about that. So a little bit of background on what we do. So we're a biotech company leveraging quantum computers to find novel drugs. So let's start off with a very simplistic example of the world, protein ligand interaction. Here, the word ligand is chemists speak for small molecule. So we're showing here the SMYD3 protein, the white surface, so what it looks like in 3 dimensions. And you'll notice there's a green region that's highlighted. That would be the binding pocket. So that's essentially a groove, in this case, a groove, where a small molecule can fit in and embed into the protein. That small molecule would have some therapeutic usage. In cancer, you might be trying to kill a cell. You might be trying to upregulate or downregulate some other process in the body through the use of this small molecule. These projects are typically involve a small binding pocket. Small is a relative thing. I mean, we are talking about proteins, but we're not talking about large biologic molecules like Ozempic or any of those other wonderful biologics or immunotherapies. This is very classical small molecule like aspirin, for example. So more complex use case that's one of the most challenging areas in drug discovery would be protein-protein interaction. Here we see 2 different proteins, one in lighter gray, one in darker gray, and we're highlighting an interface between the 2, the yellow side and the red side, where those two proteins are going to come together, and in this case, cause Ebola, which I think we can all recognize is not a good circumstance, and we want to try and inhibit that interaction so that Ebola doesn't come forth in the human body. So in general, these kinds of cases involve a larger surface area, so as opposed to the small binding pocket shown in green, you're looking at a larger surface area that you need to make contact with, as well as there's rarely circumstance where you have a molecule already bound in that interface, so you don't have a template that you're working from. So again, to reiterate, the goal here is to block that protein-protein interface. So we want to design a small molecule shown in the middle that fits in roughly where that fuchsia arrow is that will drive the proteins apart, or better said, they can't come together because there's already something sticking there in the middle. This work was all done under DARPA impact contract and leverages work that was published in Cell and focuses on the Niemann-Pick C1 receptor, or protein. So we see that in fuchsia here, and the salient thing to understand about this particular image of the protein is we're identifying 3 different spots on the protein we're labeling. One is the base, which would be a region of the protein where you can anchor a molecule. Whoops, back up one if we can. Maybe. Uh-oh. There we go. There we go. All right, we're back on track. So we've got 3 different areas: base, the interaction, the hotspot interaction, and hotspot have specific meaning for computational chemists that aren't highly relevant here, but they do denote areas where we want to make sure that we are interacting with the target protein in PC1 to block Ebola, the green ribbon, from actually touching the protein and binding to it. Maybe I've got control. I don't have control. There we go. All right. So step 1 is we need to explore the binding pocket of interest. So that's what we're sort of visualizing here. That way we can characterize the pocket in terms of total volume of the pocket, volume of areas of the pocket, be able to identify hydrogen bonds, donors, hydrogen bond donors and acceptors, and other key characteristics. The goal here is to be able to characterize it so that when we look at a very large molecular space, 10 to the 30th molecules, we can sample that to get the right fragments out to be able to design novel molecules. There we go. So when I'm saying fragments, we've got, so in the prior slide, we talked about being able to describe the binding pocket of NPC1, and we come up with a template molecule. On the left-hand side, we see a whole molecule that's been divided up with the little gray boxes into separate R groups, is what it's called in computational chemistry. And these are just different fragments of the whole molecule. Once we have described the binding pocket with regard to whole molecule, break that whole molecule up into discrete fragments, we can now go to the large library of fragments and gather them together to make a large combinatorial library that will permute over on the quantum computer to be able to generate whole molecules. So step 1, shown sort of in the right of center of the screen, is fragmenting the whole molecule. Step 2 is creating the library of fragments that we're going to permute over. Step 3 is then going to be assemble those into whole molecules that could become drugs. So in total, this represents a constraint satisfaction problem, which in our case is order factorial complex. It's not strictly mathematically true, but order of factorial is a good way to understand the complexity of this problem for the permutation. So our primary objective is going to be finding a combination of these fragments that when we put them together have the highest pharmacophore score. Pharmacophore score here is roughly a fitness metric that characterizes how good the molecule is at binding to the binding pocket. Is it got appropriate drug-like characteristics? So on and so forth. So from that objective, we have several constraints that must be honored. So we've talked about dividing it up into separate regions, R1, R2, R3. Each region must have exactly one fragment, not zero and not two. Sometimes the quantum computer will give you nonsensical results that have, zero fragments or two fragments. That's not a valid satisfaction of the constraints. There's other constraints that are necessary, such as we talk about hydrogen bond donors and acceptors. And lastly, fragments all have to be able to connect. We have to be able to assemble a whole molecule. If R2 and R3 can't actually connect, we don't have a whole molecule. So yet another example of a constraint we need to honor. When we consider in this work that we did for DARPA, what exemplars there are of solving constraint satisfaction problems in the wild, certainly we consider brute force approaches. That is a valid solution for very small take-your-toy problems. But in drug discovery, when you have 10 to the 30th molecules, you're quickly going to run out of resources. Oops, there we go. More conventional approaches would be some form of sampling, so simulate annealing or greedy steepest descent, which were the primary ones we're going to consider in the results. Next slide. Maybe. I do not have control. There we go. Great. Thank you. So we do have in production a QUBO-based solution, so quadratic unconstrained binary optimization, that when we said high pharmacophore score, in this case, it's looking for low energy solutions. So you essentially flip the sign of the pharmacophore score, and that's what we're using in production today. In this body of work, we were exploring alternative modalities of solving optimization problems, looking at both BQAs as well as subtype of quantum approximation optimization algorithms, apologies, QA/OA, to align with the combinatorial nature of the libraries we're examining. So I'd highlight here that part of this work was done using BlueQubit's quantum simulator, where we were looking at a small number of fragments to vet out our solution to be able to go back and forth and rapidly iterate with our computational chemists before we actually deployed the final solution to IBM's Eagle processor to satisfy the contract. The BlueQubit simulator was very helpful in rapidly iterating for our development cycle. So really appreciate the opportunity to work with them. Next slide, please. So let's look at some results here. So in this slide, we're looking at a particular library, R177. So in this particular experiment, the whole molecule was divided into 3 regions, not into 4. And 99 fragments were used. I believe that was 33 fragments for each region, but please don't quote me on that. But again, order factorial complexity when you're permuting over those different things. So looking at the far left, we see the results from simulated annealing. While it provides a nice pause, in drug discovery, you need a wide variety of potential molecules because some molecules may be toxic, some may be not synthesizable, whole variety of things when we actually get to the real world and actually make molecules. So we want a nice distribution of molecules for downstream processes to handle. Looking at, again, the classical approaches of simulated annealing and stratified random sampling, while they did provide nice coverage, their average pharmacophore score relatively low compared to what we need to target. If we look at the D-Wave Advantage, while it provided a nice spread and was at the higher end of things, it wasn't actually able to reach the most optimal pharmacophore score seen in the data set, which was seen using the QAOA solution on IBM's Eagle processor. And again, the QAOA solution was able to provide the broadest coverage of solutions. It's not the conventional way you would think about solving a problem, that you want lots of potential answers. But in this circumstance, yes, you do not want the single best. You want a wide variety so that you can explore different hypotheses and come up with really the best result. Next slide, please. Another way of looking at this is looking at aggregated results over multiple runs. So here we're comparing simulated annealing to QAOA, and you'll see that simulated annealing rapidly is trapped in a plateau and not able to find additional solutions, whereas QAOA is able to continue finding novel solutions that is linear with respect to time, which is very important. As we can see in this circumstance, we're looking at 99 fragments. We regularly look at libraries that have hundreds or thousands of fragments. So being able to look at a large chemical space allows us to move forward and find novel molecules that are actually usable, functional, synthesizable molecules. That is a problem with some other computational approaches, is they may describe a good molecule, but it may not actually be synthesized. So really happy about that set of results. And I think we've got one more slide to go. Yes, so some conclusions. Top line conclusion is both annealing and gate-based systems yielded superior results than classical methods for this data set. There's been mentions earlier in this presentation about quantum advantage and supremacy. We are in the software business. We are in the drug discovery business, not in the quantum hardware business. Other people get to make those claims. We simply find quantum methods and quantum systems to be efficacious for our goal of finding novel drugs. Another important thing to highlight again is our QAOA implementation was able to efficiently explore that solution space and wasn't impaired by the localized plateaus on the Pareto front. The energy landscape, the Pareto front, is very bumpy, shall we say, and classical solutions are prone to getting trapped. And in such a large solution space, it's very important to be able to continue to explore that energy landscape. Final thing I would comment is that as quantum computers continue to grow in size as a function of the number of qubits, QAOA will continue to efficiently explore large solution spaces. So really happy about those results from this work. I'd highlight that this work and results were developed by Mr. Maurice Benson, who I think is in the audience with us today, as well as Dr. Anna Petrov. And this work was funded by DARPA's IMPACT program, which we really appreciate their support. And with that, I think I'll hand the baton back.
Perfect. Thank you, Bill, and thank you, Haik, for a really good presentation. Yeah, hope it will spark some discussion on the topic. There's actually a few questions we can cover. So I see one on a Q&A section, but feel free to add more questions if you have, guys. We have a couple more minutes before the end. So I'll start out with the first question. How do you compute for pharma score? You need to compute it for each combination of fragments proposed by the optimization algorithms. Is that correct?
So it's taking a wide variety of characteristics for each fragment, and in the on the quantum computer, it's, for the sake of argument, summing those back together to create the aggregate value. There are some values that are not additive in nature and can only be computed once the entire molecule is created. That happens at a later stage on classical systems. Unfortunately, we're not able to do full scale solving the Schrodinger equation on current quantum systems, but it does provide us the nice advantage of looking at very large combinatorial libraries.
Great. Next one, yeah, I'll go quickly through the questions, guys. Also, I think related to the pharma aspects, can this system screen molecular complex conformation changing before and after the molecular binding?
Unfortunately, that's not a question. I think I know what's being asked, but I am a software engineer, not a computational chemist. So be more than happy to follow up on that. Please send me an email and I will get it routed to the appropriate powers that be.
Perfect. Great. I'll bring up one more question. I think this might be directed more to Heike on a kind of implementation stuff. Was the QAOA completed on a quantum device or a simulator? Simulated.
So on BlueQubit platform, we have both simulators and quantum devices. I think Polaris has been using mostly the simulation capabilities of the platform, but some of our users are seeing a workflow where basically first they run simulations and then they take the best algorithms and then run them on quantum hardware.
Perfect. Thank you, guys. Any last minute questions? I think we have 1 more minute until the end of the webinar. Yeah, I don't see anything else. So I think with that, let's wrap it up. Thanks again, Heike and Bill, for the presentation.
I'll
And
just add,
please, yeah,
yeah.
go ahead. Yeah,
Yeah, I'll just add that, yeah, if anyone has any more questions, please feel free to email me or Bill about the webinar, about the content, or just about Polaris or BlueQubit. We'll be happy to follow up.
fantastic. Excellent. Thanks again for joining. Hope this was helpful for you and look forward to continuing the conversation.